Okay, so in the last video we talked about inoculating McConkey agar. That's always going to be your first step to get your gram-positive separate from your gram-negative unknown. Another series of tests that's useful for differentiating between gram-negative rod-shaped organisms is the IMViC test. Okay, so that's a series of 4 different test tubes that typically are done together, but they don't really necessarily have to be for any reasons. It's an acronym, and we're going to talk about each one of these tests separately, but the I stands for Indole Test, M for Methyl Red, V for Vogues Proscower, and the C is for citrate. The I is just added for ease of pronunciation. All right, so we're going to talk about all 4 of those tests, and we're also going to talk about inoculating a urea test today. All right, so let's start with the I for the Indole test. Okay, Indole is actually the product of the reaction that we're testing for here. Okay, we are looking for organisms that have an enzyme. I always have trouble spelling tryptophan, that can break down the amino acid tryptophan if they have the enzyme tryptophanase into another amino acid, indole, plus pyruvic acid, and a little bit of ammonia. Okay, so for this particular test, I think it's useful to look at the structure here. That kind of helps us understand what's going on. I don't have this memorized, and I wouldn't want you to spend the time doing that either. But here's the structure, if I do it right, of the amino acid tryptophan. Okay, so this enzyme, tryptophanase, cleaves the molecule about right here. Okay, and then what we end up with here, this 2-ringed structure here is actually the indole. Whoops, pretend that's one bond there. Okay, this is the indole, and then 3-carbon pyruvic acid. Okay, let's see, I don't have something quite right. Okay, sorry, wrote this wrong. It's actually CH3 here. Forgot an oxygen, and I was counting my oxygens and it didn't come out right because actually there's some water in here. Okay, and this is ammonia or ammonium NH4. Okay, one or the other. So, oops, sorry about that. Anyway, a lot of trouble for this is the reaction that you should be familiar with. Okay. What we test for is actually the indole. We have a reagent that we're going to add here. It's called Vogues reagent. Okay, and what we're looking for, you can come in and add that after it's incubated. And what we're looking for a positive result here is a nice red ring. Okay, so you've got a broth tube with broth that's been enriched with tryptophan. Okay, you'll inoculate that like you do a normal broth, and then that Vogues reagent, you're going to add about 5 drops, and really just want to lay that on the top there. You don't want to shake that up at all because we're just looking for that red ring to be sitting right on the top. Now, I don't know what this is going to look like this year. Typically, the Vogues reagent is a yellow color. Okay, so if we have a negative indole test, it's going to look something like this after you add that Kovacs reagent. Or this may be an uninoculated tube, I'm not sure, but that yellow Kovacs just sits right on top there and you can hardly see it. Now, last year, the Kovacs reagent, the way we made it up, it was kind of brownish to begin with, so the negative result looked kind of brown. Okay, the positive result, it needs to be red. Okay, and it's really pretty obvious when you see it, but if it's a little brownish, okay, brown is not red. All right, so make sure we see a nice red ring there, and we'll look at that pretty carefully. But usually this works great. 3 of our organisms are indole positive, and the other 7 are indole negative. Okay, the next 2 tests in that IMViC series are a little bit more complicated to think through. We're going to talk about them together. It's the Methyl Red test and the Vogues Proscower test. Now, both of these tests are going to test for fermentation pathway. All right, so all of our enteric organisms, okay, so that's of that list of 10, 8 of them are considered enterics. They all, are going to ferment or by glycolysis convert glucose to pyruvate. Sorry, can't tell the difference between my U's and my V's here. Pyruvic acid. Okay, and then the fermentation pathway that they use at this branch point, pyruvate, is either one of 2 ways. Either they're going to do a mixed acid fermentation, or they're going to be able to do a 2,3 butane diol fermentation. Okay, so all of our enterics either do one fermentation pathway or the other. They don't do both. Okay, so if they do a mixed acid fermentation pathway, I'm not going to go through that whole pathway, but the end products, like you may suspect, lots of different acids. So we get things like lactic acid, formic acid, succinic acid, and acetic acid. Okay, and there's typically some gas that comes off of there, carbon dioxide, but the pH here is pretty low. It's about pH 4. So it's really easy to test with a pH indicator. Okay, we're going to add, again, the pH indicator Methyl Red. Okay, thus the name of this pathway, thus the name of the test, the Methyl Red test. Okay, and again, that tests for a mixed acid fermentation pathway. For a positive result, we get a red color change. For a negative result, that red pH indicator will really quickly turn yellow. All right, I don't have a number as what the pH needs to be, but this will be red if it's positive, and again, if it's a little bit more basic than that, it will turn yellow. Okay, the 2,3 butane diol fermentation, with this pathway, there's a couple of intermediates that I want to mention, acetoin and diacetyl, and then the end product is 2,3 butane diol, and thus the name of the pathway. Okay, to read whether the organism uses a 2,3 butane diol pathway, you're going to add a couple of reagents that we will call VPA and VPB. Okay, so VPA is actually alpha naphthol, and VPB is potassium hydroxide. But A and B are good enough for me. And those reagents actually react, whether you say it reacts with acetoin or diacetyl, both is really technically correct. The acetoin actually oxidizes to diacetyl, which reacts with the reagents. So whichever one you say the reagents react with is both correct. You're looking for kind of a brownish red color change here. And it's really important, make a note here, you want to shake that broth tube when you add the reagents, because the VPA and the VPB react much quicker with the diacetyl in the presence of oxygen. All right, so if you just add those reagents, it's like, what is it, like 3 drops of one and 9 of the other, something like that. Make sure you follow along whatever it says in the book. If you just let it sit, it takes about half an hour to react and to get any color change. But if you shake it up, again, kind of loosen the cap and then tighten it again when you shake it up, that will let some oxygen in and it will react a lot more quickly. Okay, so here, this is the Vogues Proscower test. Okay, I guess give a nod to the guy that developed it, most likely. Okay, so we have the Methyl Red test, testing for the mixed acid fermentation, and the Vogues Proscower test, which tests for the 2,3-butane-diol fermentation. Okay, the media that you inoculate, you're going to inoculate 2 broth tubes, and you're just going to label them both. Well, the media is MRVP broth. Okay, so it has kind of a lower volume in it there. You're going to inoculate 2 MRVP tubes, and even though you're going to inoculate them the same way, you're going to label one your MR tube and one your VP tube. And that will remind you that after incubation to add the Methyl Red to the MR tube and the VP A and B to the VP tube. And that's after incubation. Okay, so some examples of what that looks like. Here's a positive Methyl Red test. Okay, bright red. It really has to be red. Here's the negative test after you add those Methyl Red drops. And for the Vogues Proscower test, remember, you got to shake this one up a little bit. Here's a positive. It's often kind of a dirty looking red, but it does get more red the longer it sits. I would give this one a good 15 or 20 minutes before you for sure call it negative. Sometimes it's just slow to happen. Okay, and here's a negative, again, VP test. Okay, so like I said, all of our enterics, organisms in that family, Enterobacteriaceae, will be positive either for one pathway or the other, not both. So if this Methyl Red test turns positive right there, don't knock yourself out shaking this over here, because that's not going to be positive. Okay, but remember, we have 2 organisms, and that's actually Pseudomonas and Alkaligenes, that are not in that family, Enterobacteriaceae. They're not in that family because they don't ferment glucose at all. So they're not going to do either pathway. All right, so positive for one or the other, or neither. But you won't get positive for both. All right, so moving on to the 4th test in that IMViC series, that is the citrate test. Okay, this is our first slant that we're going to inoculate, and we're testing to see if organisms can use citrate, this is a 3-carbon compound, as their sole carbon source. Okay, they can if they have the enzyme citrase, which is actually a permease enzyme that allows them to bring that inside of the cell. Now, the end products here are rather complicated. One of the products is ammonia. And when you see ammonia, you should think alkaline. All right, so Really, that's the key, is that we're looking for alkaline end products. Okay, our pH indicator in the slant is bromethymol blue. Okay, this is one that you haven't seen before. It has a neutral pH. It's green. At alkaline, it's going to be kind of a nice dark Prussian blue, and acidic, it would be yellow. We're only going to see it at either green or blue, though. Okay, so you're going to inoculate your slant. Remember, just rub the organism all over the slant. And then, very important when you inoculate is to keep the cap loose. Organisms that can utilize citrates are strict aerobes. So they need to have that oxygen exchange under the cap. So you don't want it so loose that it's going to fall off, but fairly loose. If it's cranked on there tight, this will not work. All right, so leave the cap loose, and for a positive test after incubation, we don't need to add any other reagents. We're looking for a nice Prussian blue color change, and sometimes that's just on the slant portion here. So make sure you look at that carefully, turn it to the side. If you see a bluish color change, that's a positive. And here's a negative, that Simmons citrate agar, that's what we call the media, is green to begin with, and this is basically an uninoculated tube or a negative tube. Okay. Sorry, correction. And this didn't look like, this is a 6-carbon sugar. Not that you needed to know that, but just to get my facts straight here. Okay, so lastly, let's talk about the urea test. This is a test that's not part of the IMViC series, but it is, again, helpful to differentiate between gram-negative rods. Okay, we're looking for organisms that can, let me write this up here, the urea test. We're looking for organisms that or organisms that can produce urease are able to neutralize acid in the stomach. For organisms that cause ulcers and can live actually in that low pH environment of the stomach, like pH 2, if they can produce urease and kind of break down this waste product urea into some really alkaline end products, and they can kind of make a little environment for themselves that's not so acidic. Okay, so what you end up with here, NH3, okay, again, ammonia, okay, and in the presence of water, which there's lots of in the urea slant, the ammonia and carbon dioxide combine to form ammonium carbonate. I'll write that here. Okay, and important thing there is that that's quite an alkaline end product. Okay, so we have a pH indicator, Phenol Red. You're going to see that indicator again in the next lab in exercise 15. Lots of pH indicators today. I usually tell students, know the media, know the reaction that we're looking for, you know, like the substrates, the products, the enzymes. Know that there is a pH indicator in there, but whether it's neutral red or phenol red or methyl red, I'm not so much worried about that and would not ask you that on an exam. Okay, so with phenol red, basically with that indicator this time, let's see, alkaline is kind of a hot pink color, and acidic we get kind of a yellow color change. Okay, and it's kind of orange when we first inoculate it. My uninoculated tube for some reason got turned kind of wonky, sorry about that, but it's kind of a orangey color when you inoculate it, so like the citrate test, you're going to just inoculate the slant here. And then we're going to incubate it. No other reagents to add. For a negative urea test, we're looking for a yellow or orange change. So it might change a little bit yellowish, or it may not change at all from that kind of tangerine orange color it was when you first inoculated it. And a positive urea test is going to be a hot pink color change over here. So it may just be the slant that turns hot pink, or it may be the whole thing if you've got an organism that really makes a lot of urease. Okay, here's some pictures that I just got off the web. They're a little better pictures here, but here's an uninoculated tube, what it looks like to begin with. You're going to streak the surface here. I should have mentioned also for this one, leave the cap loose after you inoculate it. That air exchange under the cap there is really important for the reaction to take place. For a positive result, you may have the whole tube pink, or maybe just partially, maybe the slant, for example. And here's a negative. A couple of our organisms, we actually see it turn a little bit yellowish. Okay. So it's a lot of tests to kind of keep straight. You'll want to kind of keep going over it just so it gets a little bit more familiar. The good news is we only have 2 more tests to inoculate, and then you'll be able to discern between all 10 of those gram negatives. All right, have a great day.